Mark Janender, and I'm the president of the AFHU Western Region. It's my honor to welcome you to another fascinating innovation series webinar. Thank you for joining us today to hear Professor Simon Benita, head of the Laboratory of Nano Delivery Systems at the Hebrew University. I'd like to begin by sharing some exciting news. The Hebrew University recently announced that they are establishing a new center to address one of the most pressing issues of our time, the Hebrew University Center for Climate Science. The center will foster ideas, research, and interdisciplinary brainstorming around the climate crisis. This is just one example of how HU is actively working to make the future better for all of us. In addition, HU has opened the first phase of the new Biosafety Lab Level 3 for advanced study of live coronavirus. This lab is the only one of its kind in Israel for non-government research, and HU is making it available to all universities. This is a game changer in accelerating coronavirus research and will also prepare HU for future research and viruses. Another example of the important research being led at HU is the important work of Professor Simon Benita, the former director of the Institute for Drug Research and the former head of the School of Pharmacy at the Hebrew University. Professor Benita's work is more than impressive. He has published 160 research articles and 20 book chapters, edited or co-edited 5 books, and been issued 38 patents and patent applications. Professor Benita has developed a drug delivery technology which increases the bioavailability of drugs to dissolve in fat or lipids, which had previously been poorly soluble in water, and thus have limited bioavailability and clinical efficacy. A bit of a mouthful, but I'm sure we'll learn a little bit more about this throughout the speech, the presentation. He has founded several companies based on his work, such as Macrobea and NanoBioSim Limited, also known as BNS, which focuses on innovative nanocarriers as a solution of choice for enhanced topical drug delivery to improve delivery profile and therapeutic performance. Another mouthful. I'm especially happy to have Professor Benita with us today. I'm being told this is the first Zoom call that he's accepted. He and I have gotten to know each other as I'm an investor in BNS and have been involved in that company for over a year and have been tracking its progress. So I know how impressive the work is. I don't know how much he's going to go into some of the detail of the applications at BNS, but I'm going to ask him to at the end so we can all learn about some of the exciting research being done there. We're just really privileged to have you here. Thank you very much for making yourself available. This doesn't replace, even though we're very fortunate to be your first Zoom and we appreciate it, this doesn't replace the invitation to come see us in person, which we'd like to do when travel allows. So it's my pleasure to welcome you and thank you for joining us.
Thank you, Mark, and I will share with you the screen so you can see my presentation. I hope that all of you are seeing it.
Yes, we are good.
Okay, you are good. I just will initiate the pointer. Here it is. So, Mark, thank you very much for your generous description of my work. And I am very pleased to be with you and to deliver this lecture. I feel that I am close to LA. This is the technology, and I really would prefer to be in front of you and discuss and have the contact with the audience. But these hard, difficult days push us to do things overthink. What I am going to speak with you today is how we can improve drug performance thanks to biotechnology. Just for you, I start with something that you probably will understand. This is a comprehensive map of FDA-approved pharmaceutical products. In fact, the global pharmaceutical market in the world of drugs today is more than $1.25 trillion. What you have here is a pie about the repartition of a segment in pharmacy in the United States published 2 years ago. And you can see that the pharmaceutical market in the US is dominated by the oral delivery oral route of administration. The second is injectables, and the third is dermal. These three segments, in fact, are more than 93% of the pharmaceutical drug market in the US. The second thing that you need to know, in 2017, the US market was accounted for more than 40% of the global pharmaceutical market. Today, according to what I read in the internet, it is almost 43%. So everything that you see here about dosage forms and how the drug are administered to the populations and to the subject is reflecting the global market in the world. Now, as all of you know, the oral route is the most preferred route of administration. And this is obvious because it is easily self-administered. You don't need to go anywhere. Relatively cheap, and also you have no systemic infection complications as we have with the injectable segment. And the injectable segment, as I told you, is large today. And this happened because of the development of immunotherapy and all the proteins and all the biologics drugs that came to the market, antibodies, monoclonal antibodies, proteins, etc. Now, it is the easiest way to take a drug as we are eating and anything else. And what you have here, and it is very important, is despite that everything which penetrates the body is going through the mouth, mostly, it is a very complex system. Very complex. First, when you swallow something, and a drug can be in a tablet, in a capsule, in a solution, etc. First reach the stomach. You have here the stomach, and the stomach is large. In fact, in the stomach, you have a very hard environment which is acidic and can destroy many, many drugs. And this is a problem, and we need to protect them because nothing almost is absorbed through the stomach. What happened, we have a transit from the stomach, and relatively in less than few minutes, depending on the dosage, if you are not fast, then will go directly to the duodenum, which is the connection between the stomach and the small intestine, as you see here, the duodenum. Now, the duodenum is in fact the organ or the part of the organ that absorbs most of the drugs that we swallow, okay? And here the pH is about a pH of the blood and more or less, and therefore here, but we have a problem. Nothing can be absorbed in the cells through the membranes of the organs of the cells if it is not dissolved in the gastrointestinal fluid, in the lumen of this tube, as you can see here, the intestine. So it needs to be dissolved as you take salt and you put it in water and it becomes a solution, then you can absorb the salt. And here is the same. If it will remain as a powder, nothing will happen. So when you take a solid tablet and a capsule and you swallowed it, if the powder will not disintegrate and the drugs will not be extracted by the fluids from the compounds, the powders, nothing will be absorbed. Now the second and most complicated, and we have here one of the most sophisticated machines that you can see how we do accept and delivered nutrients, anything, nothing that goes through the oral route will directly reach the blood. And this is something amazing, because you can't believe that through the intestine, it can go to the blood. No, you have here a very important portal, what we call the vein, the portal vein. It is in the liver. Anything from the stomach, intestine, from the colon and anything else, everything you can see the blue here, capillaries everywhere, anything is collected and moves through the ramifications to the portal vein directly to the liver. And the liver is in fact the first organ in the body encountering anything that we do absorb. And here, this is the most, maybe one of the most important organs in our body. Is objective and function is to detoxify potent, dangerous compounds identified by the body. So the drugs, when they can reach the liver, most of them may be degraded. The liver is full of enzymes, of macromolecules, degrading and metabolizing the drugs. And some of them are degraded before reaching the blood. So do not imagine when you drink something or you eat something or you absorb a drug of 100 milligrams, for example, all the 100 milligrams of this drug will reach the blood. There is nothing like that. It depends how much the drug is sensitive and half of it will reach the liver. Now, before reaching the liver, which is the ultimate organ before reaching the circulation, in the intestine, in all these billions of cells of the gut, there are additional mechanisms of protection. And this is a very complicated system and mechanism, and it took many decades to understand everything. And there are two more additional mechanisms. One, which is in fact a pump located in the membrane of every cell of the intestine. The cells in the intestine are called enterocytes. These enterocyte cells have in their membrane what we say guards that will prevent, and even if molecules are absorbed into the cells, there is a glycoprotein here in the membrane. If it recognizes the cell as being really a danger or at risk, they will efflux back the drug. So this system, in fact, is one of the filters of the body to prevent toxic nutrients or toxic drugs to reach the cells. Now, most of the drugs, in fact, that are absorbed through the GI tract will first, if they pass the pump, as I told you, and into the enterocyte, there are also some enzymes degrading, they will reach finally the liver. And here is the final mechanism of defense before entering in the blood. Now we have a second system. 99%, even more, of the drugs follow this mechanism. All of them reach the liver, and this is why we defined this system, the hepatic first pass effect. Nothing penetrates the blood without moving from the liver, except about 0.5% of the nutrients, and these nutrients are, as I told you, to be absorbed, you need to be dissolved in the gastrointestinal fluids, and they are mostly aqueous solutions, dispersions. It is aqueous. So most of the poorly aqueous drugs are difficult to be absorbed. And you can ask me why we are increasing in weight if you are eating fatty food, lipids, oils, and anything fatty. In fact, they are not dissolved in oils, in waters, as you know, and they should not penetrate the body. However, our body needs these fatty elements because they are essential to our survival, and they are needed by many organs in the body. So I will not go inside, and this system is called the lymphatic system. In fact, the lymphatic system is a parallel system to the blood circulation, and its functions are first to maintain the body fluid balance, to remove the macromolecules from the interstitial fluid. In fact, it is evacuating all our degraded metabolism and waste into the colon back out of the body. And finally, it is this system which is in charge of absorbing the lipids, the fatty compounds. So if we have very fatty compounds, when they enter into the enterocyte, these fatty compounds are subjected to some transformation, and these small lipid droplets about the size of 100 nanometers that we call chylomicrons. Chylomicrons is from lymphatic microns, small particles of the lymph. They bypass all the system, and through the lymphatic system, they go directly to the blood. So we call such a system bypassing the liver. So remember, it's very important. Now, as I told you, we have many important and potent drugs that are needed for our survival: antibiotics, antivirals, antifungal, chemotherapy, immunosuppressant drugs. All of them are defined by our body as xenobiotics, meaning they can be harmful to our body. So because of that, the pump that is at the interface into the membrane of the cells in the gut, they will prevent from these actives. Once they are inside the cell, they will be effluxed and expelled back because the body does not want them. And you know, and all of you know, that once you are in the blood, every drug that reaches the blood in seconds will irrigate all the organs of the body. We have other very specific organs. I don't want to speak about the blood-brain barrier. Our brain is also protected. In addition to all what I am saying here in the intestine, we have in the brain, which is the most vital and critical organ for us, a barrier preventing from anything to enter without a transporter. So the brain takes what it needs to take. And this is the most complicated organ. And I don't want, so speaking here, in fact, you have to understand that potent drugs do very good things and can relieve a disease, but also goes everywhere in healthy organs and can elicit side effects. And we need to understand this. And the problem is benefit ratio risk. This is always like this in the, nothing comes with good without a little bit of bad. Now, where is the problem? More and more, at least 50% of the new drugs and about 25% of existing drugs suffer from such mechanism. And then this is why when we speak about oral delivery, we speak in fact about bioavailability. Why? You never know how much of a drug will penetrate from oral administration and reach the ultimate site, which is the blood. We measure the concentration in the blood because once it is in the blood, it can go everywhere. So every oral administered drug has an oral availability of less of 100%. The only drug that can be considered 100% available is a solution injected in the vein. All the infusions in cancer, in chemotherapy. Anything that you receive directly in the vein as a solution is 100% bioavailable. Otherwise, even if you inject intramuscularly, subcutaneously, if you give by oral, dermal, anything, you will face a problem, something's good, of oral bioavailability. It will be limited, and some cases it will be more. So if you have a drug, and this drug reach only 20% of the amount of an absorbed drug reach the blood, you will say that your bioavailability is 20%. So sometimes it can be very difficult with such a limit. And I can tell you that FDA regards a poor absorption as 30%, 25-30% of oral bioavailability. A good bioavailability is a bioavailability above 50%, 60, 70, and you have very, very small number of drugs given orally that can reach 90%, 95% of oral bioavailability. And this is the rationale for my title. If you will improve the performance of a drug by improving oral bioavailability, yes. If you have a drug and you give it orally, and normally this drug in a simple tablet or capsule will provide an oral bioavailability of 20 or 15%, and you will succeed with a delivery system to improve this bioavailability by twofold, threefold, then you can have 50% bioavailability. And in this case, you improved the performance of the drug because a higher concentration can reach, but not too much high, to avoid toxic effect. You can reduce the frequency of the administration. Many drugs before 25, 50 years were given 3 times a day, 4 times a day, but with the stressful life that we have today, a drug should be most of the time given once daily, maximum twice daily, to respect compliance. Because once you are leaving your house and going to work, you will forget to take a drug. You will not have time to think about it. So this can be also a reason for a lack of efficacy. Now, today, we know that we have a bioavailability challenge. How we can improve the oral bioavailability of drugs? There are many approaches. I cannot cover it in 30 minutes. But I can tell you that we need to bypass the gut enzymes, as you can see here. And this is an issue because these gut enzymes are in the fluids in the intestine. They are inside the enterocyte, in the cells of the gut, and they are in the blood, and they are mostly in the liver. The second thing that you need to overcome is the PGP pump, as I told you, the glycoprotein pump, which in fact efflux outside the drugs that are absorbed. And finally, you have to bypass the liver. It's the most difficult bypass that you can imagine. And here, one of the approaches that we have used, and believe me, I didn't reach success without facing many failures. Do not believe this is a very easy issue. Nothing scams. And once something that appears today Obvious is, for researchers, it's never obvious. It's always when you learn something new, you have to think about 100 and 1,000 possibilities, and always you have to substantiate your hypothesis and slowly. And this is the reason why the development of new chemical entities to become a drug takes more than 10 years and billions of dollars of research. So one of the approaches can be inhibition of the enzyme and of the pump by other drugs. This is a good way, but a bad way also. Because if you inhibit the mechanism, real toxic nutrient can enter into the blood and make more severe side effects. And again, the ratio between efficacy and toxicity should be a benefit for therapy and for relieving the problem. So this is not a good reason. Other approaches are tested, and you need to find something that can be the ultimate delivery system. This is one of our techniques that we have developed 10 years ago. And this technique we learn here is a Trojan nanotechnology. In fact, after publishing in the literature that inhibiting the mechanism of defense will not be beneficial and maybe even worsening the issues, other people tried other things, nanotechnology, nano things. The problem with nanotechnology is that we have very small coating in the size of 100 nanometers, which is very, very, very small, and you cannot see it through your eyes. And just to give you a comparison, we need really huge electronic microscopes to see the particle. They are in the size, we are 100 times more bigger than an erythrocyte. This is a blood cell. So they are very small particles. And these particles cannot stand and coat the drug inside once you are giving them by oral delivery, because first they will encounter the gastric harsh environment of the stomach and they will be destroyed there. Even if you will protect them, they will maybe destroyed in the intestine. So what we had in mind at that time is to use the Trojan horse approach that was used 100 and even many thousands of years. The Trojan horse is what we have done here. We took here an oil core where the drug is dissolved here, and we coated it with a polymeric envelope. So you have nanocapsules, and we embedded these nanocapsules in a microparticles, as you can see here. You can, in fact, enter here thousands of small nanoparticles, and these nanoparticles are enterocoated, meaning they will not open before reaching the intestine. In the stomach, they will remain closed and protect the nanoparticles because they are resistant to the acidic medium of the stomach, and only when we reach the duodenum, then they will open. As you can see here, this is as it looks like in reality. You can see that we have a microparticle estimated here to 10 microns. We have here, you can see a scale with 2 microns. What we did here, we cut with a scalpel, and we were fortunate to see inside of a microparticle. You can see here hundreds and dozens of nanocapsules here. This envelope will open only in the jejunum, in the intestine portion of the jejunum. And I will show you here something that will be important to see. You can see this Trojan drug delivery system. These microparticles penetrate through the the stomach. They cannot be opened. Only in the duodenum, where the pH change, and in the jejunum, they open, and then the small nanocapsules do penetrate through the intestine. And since they are oil droplets, like the normal triglyceride, natural triglyceride, in the enterocyte, they are transformed in chylomicrons and are taken from the enterocyte through the lymph nodes in the lymphatic system, bypassing the liver. And they are directly reaching the blood. This is really something that we patented it, and for the first part, it was the first time in my life when we submitted a patent application. They gave immediately the patent without any remark. We were amazed to see this thing, because normally you will have many remarks and things to correct before your patent will be approved. Here it was approved without any remark because it was totally new. It is patented worldwide, and this is a patent that we have in BNS, of course, and I will explain this in a little bit. Just to give you an example, because you know about all the cannabis, and you heard about cannabis in all your states. In California, it's very famous. CBD and THC are given in everywhere, in any state, in any country. The oral bioavailability of CBD and MCT and THC in these oil solutions that you can acquire in the dispensers are in fact with the oral bioavailability of 6%, meaning that 94% of the cannabis actives are wasted. And in this kind of formulations, in the Trojan, 2 formulations, now you have here a ratio between CBD and THC. It doesn't matter. And you have the CBD in MCT oil solution. I am going to spare to you the calculations of oral bioavailability, but what I can tell you is the oil solution gives 40. One of the formulations here gives 400, is 10 times more. And here, almost 700, which is 17.7-fold more, meaning that you can increase almost to 100% your bioavailability. So we are short in time. I will tell you a few things that Mark mentioned. We have BioNanotechnology. This is a company that I created in 2016 when I retired officially from active professor, but I am not out of the university. On the contrary, I devote all my time to the Hebrew University since 2016, as you can see, even today. And many of my technologies that I keep for me for the last part of my life, and which is also the most important part, because in contrast to sportmen's, in science, as you are getting older, you have much more experience and you know better. And also you learn to know that we have still a lot to learn and we cannot be boring. It is a very exciting life. And we have exclusive technologies portfolio about 5 to 6 patents. And Yisung, the TTO company of Vehuji, is a co-founder of BNS and a major shareholder. And we operate in 4 verticals. I am going to give you these verticals here. We have a medicinal cannabis, ocular delivery, topical delivery, and oral delivery. All these forms is to improve the oral bioavailability, not to improve the efficacy, but to reduce the concentration and to reduce the numbers of frequency. Instead of 3 times CBD per day to give only 1, instead of 700 milligrams to provide 50 milligrams, which is a huge saving in economical situation. And the second one, these two activities is ocular and topical nanocarriers, again, nanotechnology to improve the penetration of various drugs, as you can see here, in atopic dermatitis, in anti-infections, and here for inflammations in the eye. Tacrolimus is well known. It is a drug that prevents the rejection of transplanted organs, and it is also one of the most efficient drugs for inflammatory diseases in the eye. And today, for this issue, we give only steroids and huge amount of steroids, and this put at risk the persons because of cataracts and glaucoma, etc. And close to it, we have peripheral CB1 receptors, nothing to do with cannabis, because we work here with other things, and this is new chemical entities that we are using. And in fact, we are developing drug for obesity and other metabolic syndrome. And needless to tell you that obesity today is a very, very pandemic disease and related to many, many diseases. And we need to improve and do here. There was a drug, CB1 receptor antagonist in the market 10 years ago called Acomplia, rimonabant, and this drug was very efficient. It was developed by Sanofi, and it works very good, reduce the weight of many obese people, but has a problem. It was penetrating the blood-brain barrier, and the drug did many damages to people, depressions, etc., and it was taken out and withdrawn from the market in 2008. So today we learn much more, and we learn from the failure of other people. We are developing peripheral without penetrating the brain, and we have very nice results. And of course, OPA is an oncology program. Platinum drugs are combined in more than 50% of the chemotherapy in cancer. This is very effective drugs because the platinum is the active compound element, and in fact cross-linked the DNA of the cancer cells. They are toxic, of course, and we are using a new platinum drug, and we are having very promising results. The company is already 4.5 years active, and now we are going to reach, thanks to tacrolimus in the eye, a clinical trial in the next year. So we are close, and I can elaborate more if you are interested. Mark has my email, and you can speak with me if you want. And I am opening this lecture to your discussions and to your questions. Thank you.
Okay, let me set the video here. Thank you very much, Professor Benita. That was fascinating. And every time I listen to you, I learn a little bit more about what you're doing. I'm just trying to see if there's questions here. I'm trying to see if I can't.
Yeah, should be right in the chat to you, Mark.
Yeah.
Just okay, there it is.
Just Q&A. Okay, I will open.
Okay. So it doesn't look like I'm just looking here. I don't think we have any questions. I would like, could you spend a little more time talking about the CBD research and why the delivery system for cannabis makes a difference? I think that's something that everyone would find interesting.
Okay, I can tell you that I am working in CBD already and in THC more than 40 years. My teacher and my collaborator and my colleague, Professor Rafi Meshulam, is, as you know, the father of all the cannabis chemistry, and he discovered all the important drugs and part of the receptors. And Rafi Meshulam worked with me in collaboration on THC, and we published for the first time early 90s a paper with a nanotechnology for the eye, and we showed that THC could reduce the glaucoma. However, knowing that it is a psychiatric active drug, it was not allowed by the Ministry of Health to make a clinical trial. Since then, we worked with many drugs, and cannabis was always accompanying the School of Pharmacy, and today it is a major subject of penetration. Now, speaking about CBD, we know today a lot about cannabis. Really, we understand better. CBD is not psychoactive, and FDA can recognize it as a drug and not as a narcotic controlled drug. As you know, GW did from CBD synthetic molecules, and today it is approved for, as you know, epilepsy, rare epilepsies that it is. But we know from work that we have done in biology, in pharmacology, that CBD is a very good anti-inflammatory drug. So what we have done, we have used CBD in BNS. We did micro and micro carriers, nanoparticles and nanocapsules and nanoemulsions, and to enhance the penetration of CBD in the various tissues of the skin, in the stratum corneum, in the epidermis, in the dermis, and once they are there, it is released slowly, and we took human skin from volunteers and we stressed the skin, and once we did it, we could even follow the biomarkers, the pro-inflammatory markers, as in corona, the cytokines, that when you have an inflammation, our body releases cytokines, biomarkers that are needed to combat and fight the inflammation. And sometimes you can have over-inflammation, and CBD does decrease the concentration and the release of these biomarkers, indicating that it is a very good anti-inflammatory drug. It can compare to marketed steroids. And now steroids, as you know, most of the physicians are reluctant to provide steroids over too much time. The beauty here is that our nanotechnology keeps this, depending on the formulation, the CBD in the skin. It does not go through the skin to the blood. It remains local. So you can do cosmetic products. And we did it. And this is one of the products that soon will reach the market. And we have nanoparticles, we have nanoemulsions, and we are working with companies, as you know, probably. We are negotiating, and we are trying to do things in the next forthcoming months. It's the same for THC. So today, the most advanced market, in fact, our strategy was to have a portfolio of different products that can reach rapidly the market as cannabis, so we can have some kind of revenues, drugs that can be in the market in 1 year from now, like the tacrolimus, and new chemical entities that will take 3 to 4 years more to reach the market. The initial clinical stages, before reaching the market, it will be 6 or 7 years. So we have today innovative biotechnology, science funded, based on evidence in science, that it is very important for many governments. And we are now negotiating through our shareholders and our people in the board with some of the companies worldwide. Open. I don't hear you.
Sorry, I was on mute. Thank you. So I naturally have more questions on BNS, but just to switch gears for a minute, could you comment on the research that's being done at Hebrew University around coronavirus and your thoughts on some of the research that's being done. And are there any applications for your technology with the vaccine application?
Okay,
I don't even know if that's an intelligent question. I'm just curious.
I am not directly involved in the coronavirus by myself because I am very busy with many other projects. And as you know, nanotechnology is here to improve things already done.
Right.
One of the things that we did with nanotechnology, not in the case of the coronavirus, because now coronavirus, there are two main issues that we are dealing them here in Israel. One, to find a monoclonal antibody, an antibody that can in fact neutralize or prevent the multiplication of a virus. And the other things are the vaccines. And vaccines is a totally different profession and expertise that we are. Normally, what we do here is in our Nano School of Pharmacy, we don't have the infrastructure to work with viruses. We work with delivery systems. And we can, for example, enhance the reaction to vaccines by adding adjuvants. And this is a very important issue. Now that we know that there are vaccines reaching the market, we can try to improve the immune reaction to enhance and accelerate the reaction of the formation of the antibodies. And this is something that we can be involved in. But soon we will be actors with the companies by helping them to make their vaccines more efficient. But our expertise is beyond, and it had nothing to do with the viruses. This is more a biological issue than a pharmaceutical issue for pharmacy that we are doing in Israel.
Right. Just from a bigger picture question I have is, why do you think Hebrew University has had so much success with this technology? And other than the people, I mean, is it multidisciplinary? Why is so much? Why is there? Yeah, no, I'm not talking Corona. I'm actually talking back to nanotechnology and the delivery systems.
I'm not the only one, by the way. I was lucky to be successful in many of my companies, but I also said always, in any research, you need also a part of luck.
Yes.
Saying that, the most important problem is to identify the unmet needs. The problem of many people is that sometimes they need a very comprehensive view of a field, what exists today. Don't do something that somebody else is doing in another continent and you do not know about it. So here you need people that have a very thorough and very comprehensive knowledge about everything be done. Of course, only disclosed issues. You cannot know about things that are working in labs, and there is no disclosure of anything. But in science, as we are working most of the time, we try to deliver and to expose people to our knowledge, to help humanity. We are not doing startups. We are trying, and we are professors and we are teachers. We are trying to teach people to do things. I am proud, for example, that 7 of my previous PhD students succeeded and are very rich by doing many startups in Israel and elsewhere. So what we are doing here, and this is the most important issue in our Hebrew University of Jerusalem, we have a center, very small, highly coordinated, with all the disciplines needed for the development of new chemical entities are there. And we were lucky to have royalties from Exelon, if you remember, and we did invest the money in very important things. We have created an equipment core where we have a more sophisticated equipment to do research. We can compete with any internationally and famous center of research, including the big pharma companies. And sometimes they come to see us, and how with such small amount of money, we are so successful. I have to be modest and tell you I do not know, because sometimes you start something, you never know from where you can have the obstacle. And you need to rebound. You need to face always difficulties, and we try to do it properly. And this is the reason why, and by the way, if the university is successful, it happens from time to time, and we have now a change in generation. So we are doing well. The new generation will be even better than our generation. And I am certain that a few years from now, you will see many, many innovations coming from the School of Pharmacy and the Faculty of Medicine.
We had a previous session at the beginning of coronavirus with the dean of the medical school, and heard a lot about the research, and you just basically answered what I was looking for, which is it's the interdisciplinary collaboration that seems to be part of the keys to success for how you're able to do things with more limited resource.
You're right. And we have this, we are lucky that we are all in the same site, in the campus of Hadassah, with the university, the hospital, and all the medicinal disciplines represented. And the government decided to put this lab of Corona in our site because we have the most critical mass of scientists working in biology, in medicine, in through all the aspects. If they have a problem of formulation, we are here to help them to solve it.
Yeah.
So for the time being, it is encouraging, but we are still a small country compared to the means and the funding that United States can make available to their scientists compared to Israel. You know it very well. And I think that your president did a lot and backed and supported in a very generous approach most of the companies that are today successful in the US.
100%.
If it is Pfizer, if it is Moderna, I follow all the literature. I can tell you that we are lucky that your government and the actual administration really backed them very, very strongly and support them in a way that really show the way to the other countries.
Well, look, I think that listening to your remarks and hearing this is exactly why all of us feel so proud to be affiliated with the university and supporting the research that you're doing. I have one other question, because I think a lot of people, I'm intrigued, is the collaboration with Yassoum. I think is something that I think is very interesting to people. And can you talk a little bit about how you partnered with them to launch the company?
I know very well Yassoum, the transfer technology of a university. This is probably the first one that was created more than 50 years ago. In fact, our universities always supported and appreciated what we call, in fact, applicable science. You have fundamental science and applicable science. And the beauty in our university is that both type of scientific people do appreciate one another and try to work in collaboration. So YISUM is here to protect the interest of the university and to expose the innovations of the Hebrew U faculty to the world. And frankly speaking, they are doing very well and they are helping and collaborating. Now, I do not know many transfer technology companies doing almost 100 licensing agreements per year.
Amazing.
It's amazing. You have the most international and advanced company worldwide, from Japan, from United. I see now Chinese companies, Chinese investors, Japanese investors building research centers in Israel.
Yeah.
And they came to YISUM. I can tell you, Mark, I have no free project. Any project that we start and we have preliminary data and it is presented to YISUM is sold. And now, of course, it is the same for many, many people. And we hope that, this is why I told you, that the new generation is very promising.
Right. And YISUM is holding, for a lot of the companies that they're being launched from YISUM.
In
like
fact,
BioNanotechnology.
YISUM has all the IP of the faculty, and the IP belongs to YISUM.
Right.
And we license it to the companies through YISUM. Don't misunderstand me, we are well compensated, and all of us are not working for money. We work, first of all, for publications and to remain innovative. It's very difficult today to publish publications. You know that for an academic life today, or if you do not publish, you perish.
Right. No, but go ahead.
So you need to remain at the edge of innovation. And Israel, as compared to European countries, did exploit this philosophy to make money, if possible, for the benefit of all the universities. Because the money does not go into the pocket to anyone. We improve and we give, and we improve the equipment, we build the infrastructure, we pay the fellowships. As you can understand, we are relatively a small university as compared to the big universities, and still we are not far from the 100 best universities in the world. We could be even better if we could have much more funding.
Right,
but we are doing our maximum to succeed.
Right, but I think what's encouraging is that some of these companies that are coming out of YISUM are big successes, and YISUM keeps a piece of equity, which is important because that can be reinvested. And I think that's, it's true with BNS and it's true with other companies, correct?
Any company, and we have to share. We are here to share.
Right.
And I consider the university like a family.
Right. Oh, well, look, I think it's just another example of how the school is able to do, how the university and quite honestly the country is able to do more with less. And I think it's why we're all passionate about the university and the research that's being done. And we can't thank you enough for everything you do. It's refreshing to hear someone say they're not doing it for the money, and they're doing it for the research and the publication. But I think actually what I was taken aback, what I noticed in your remarks, which is probably the most impressive thing, is the success that your students have had. Yes. And that is something that I think is really impressive. And we hope you continue to inspire more students, because that's the future, right?
Yeah, this is the best way to do. You see, when I was in the funeral on one of our biggest industrial pharmacists that died from cancer yesterday, and this is really a good scientist. And thanks to him and to others, we moved from a very, very low-tech industry 50 years ago to a very, very advanced and sophisticated and powerful industry today with billions of dollars of expert. This is really a miracle for a country surrounded by hostile people here in the Middle East.
100%. 100%. Well, you've been very generous with your time. Before I hand it over to Sherry, I just want to say thank you. It's good to see you. I'm glad you're doing well.
Me too.
It's a real pleasure for you to take the time, and we'll look forward to hearing more remarks. I'm introducing you to my newest member of my family. This is my new dog.
Yeah, I see it. He's very nice.
He's very active. He's only 10 weeks old. But I just got handed him in the middle of the conference. This is my job to puppy duty. But he's
I hope
he's a
that
very
the
friendly
audience
guy.
did enjoy about the presentation, and I will be pleased if they took with them something that nothing is easy in the nature.
100%. Well, look, I actually will get your comments. I'm going to follow up with you, and I'm going to offer up if I get questions. I may ask you to.
I can answer by email. Don't worry. I can anything. They can send me even emails, anything that they want.
No, you and I have been in touch on that, so I look forward to staying in close touch. And now I'll just to wrap it up, I'll hand it over to the Executive Director of Western Region, Sherry Coffer.
Okay.
For a few closing remarks.
Okay, Sherry.
Thank you. Professor Benita, I wanted to thank you so much for your very informative and enlightening presentation. The groundbreaking work you're doing at the university is very encouraging, and we're so grateful to you for taking the time to join us. It's really been a pleasure. Mark, I want to thank you for your inspiring and dedicated leadership. We are so grateful to you for everything you do on our behalf. And thank everybody for joining us and spending the hour with us. And we encourage you to join us for some of our upcoming webinars. And we thank you so much for your support and your involvement. Wish you to stay safe and healthy, and an early happy Thanksgiving to everybody. Look forward to seeing you soon, and all the best.
My pleasure.
Bye.
Thank you.
Thank you so much.
Have a good afternoon.
Bye. Have a good afternoon and a good night to you, Professor Benita.
Bye. Thank you.
Bye. Thank you.